So, without further ado, we're going to go ahead and get started with the endocrine system. Okay, our basic outline, we're going to kind of define what the endocrine system is. We'll talk about the different classes of the hormones, how they work, what regulates them, and then we'll actually get into the specific hormones and the glands that secrete them. So the endocrine system is kind of part of a greater scheme that controls intercellular communication. So in my last lecture, we talked about the nervous system, and that's going to be one major form of intercellular communication. And then today we're going to talk about a few more. So before we begin, let's talk about a few different terms. We have autocrine. That's a signal that's received by the cell that produced it. Okay, we have paracrine, which is based on diffusion. Okay, so a couple cells out here might secrete some kind of signaling factor, and it will diffuse to nearby cells, which would then signal them to have a certain action. And then the focus of today's lecture is endocrine, which is similar in ways in that it's based on signaling compounds, but it's also different in that in the endocrine system, we're secreting these signaling compounds into the circulation so that they can travel systemically throughout the body and exert their effects. And then signaling compounds that are secreted into the circulation are then named hormones. And one of the key points is because we're secreting these hormones into the circulation and they're going to be systemically distributed throughout the body, the effect of these hormones is going to be based on which cells possess a receptor for a specific hormone. So a hormone is going to have to bind a receptor, and that will determine which cells do what in response to that hormone. And we'll talk about that as we go. And then the key here is that intercellular communication is obviously necessary to maintain homeostasis. And homeostasis is just a state of relatively constant internal environment. And obviously homeostasis and that relatively constant internal environment is what allows you to survive. So your glucose levels have to be in a very narrow range to sustain life. Your blood pH has to be in a very narrow range to sustain life, etc. And all of that is maintained from intercellular communication using these mechanisms. So just kind of a detail thing, endocrine versus exocrine, they're absolutely not the same thing. The words come from these roots, endo meaning within, and those are compounds that are secreted in the fluids within the body. So that would be the circulation, basically the bloodstream. And those are the hormones that we're going to talk about today. And again, they're going to bind receptors to exert their effects. Whereas exocrine is totally different, and these are secreted into the external environment. So a word on the external environment, if you think about that, and you think about the human body, the gastrointestinal system, which we're going to talk about later today, is basically a hollow tube that's open on both ends running through the body. So really that's exposed to the external environment rather than the internal environment. So that's going to be the site of excretion of a lot of these exocrine compounds. So they have ducts, which endocrine glands don't have, and these ducts empty into the digestive tract, epithelial-lined orifices, and just generally the external environment. And they include sweat, oil, mucus, enzymes, etc. Okay, so back to the endocrine system, which consists of hormones. Let's talk about the different types of hormones. First off, we have peptide hormones, and these are by far the majority of hormones in the body are peptide hormones. And what they are is polypeptides that are derived from larger precursor polypeptides. And that's important, that's going to kind of be our starting point. So if we think of insulin, which is a hormone, it signals various effects in the body. It's also a peptide hormone. Insulin is actually going to come from a larger precursor called proinsulin. And proinsulin is actually going to come from a larger precursor called preproinsulin. But for us, let's start with this. So we have proinsulin, which you can see here. It's a large peptide. It's cleaved into the active form, which is insulin. And then we have a connecting peptide that we call C-peptide in the case of insulin. This is just a specific example of insulin, which is one of the most important hormones and one that you're likely to be tested on. But this kind of this idea of cleavage into the active form applies kind of generally across all of the polypeptide hormones. Okay, peptides are hydrophilic, meaning they're water soluble, and that has a couple important implications. So because they're water soluble, they can dissolve in the plasma within the bloodstream and travel freely as such, but they cannot diffuse through the plasma membrane. So the hydrophobic tails of the plasma membrane are going to exclude these hydrophilic peptide hormones from diffusing across. And because of that, peptide hormones are going to have to bind receptors on the cell membrane. So we have peptide hormones that are binding the cell membrane, but they need to get their message inside the cell. So to solve that problem, these membrane receptors are actually going to stimulate what's called a second messenger system. And it's going to induce a signaling cascade that is going to transmit the signal from the extracellular environment and the membrane to the intracellular environment, where the cell can then exert its effects. Insulin, by the way, is made by the beta cells of the pancreas. We'll talk more about that later. So first we have a question. AA should be. Okay, pretty good. So if I were to ask you a question, if I were an MCAT writer and I wanted to test your knowledge of the life cycle of hormones and how they're cleaved before they're active, this is what I would write. So a type 1 diabetic requiring insulin injections. We're measuring C-peptide concentration. So if you remember, C-peptide stands for connecting peptide, and it's cleaved before insulin is eventually exocytosed from the cell. So what that's good for in a diabetic is measuring their intrinsic pancreatic beta cell function and their endogenous insulin production. So we can't simply measure insulin in a diabetic patient because we're giving them insulin every day. So we won't be able to tell what we're giving them versus what we're making. Okay? But since we have this C-peptide, which is going to be secreted from the beta cells in the same amounts that insulin is secreted, we can measure C-peptide to give us kind of an estimation of how well their pancreas is still functioning. Okay. A couple of things that I guess I didn't give you that may make this difficult. The insulin that we give pharmacologically does not consist of C-peptide. Okay, we purify it out, so it's pure insulin. And then it's also important to know that, let's see. Based on this diagram, you may be able to figure out that every insulin molecule produced will also produce one C-peptide molecule. So they're going to be produced in equimolar amounts and secreted from the cell. So then as far as the other options, long-term blood glucose control, you may have heard of hemoglobin A1C, not really important for the MCAT. Short-term control, you can kind of take a history and you can just measure their blood glucose. And then medication compliance, again, the medicine we give does not consist of C-peptide, so that doesn't work. Okay, the next class of hormones is going to be the steroid hormones. One of the really important things you need to know about this is they are all derived from the molecule cholesterol. Okay, this is basically the parent nucleus that is modified to produce the rest of the steroid hormones. And I've given you some examples down here. So as you can see from the four-ring structure and the very hydrophobic structure, these are going to be more lipid soluble, and that has some important implications that we're going to need to know. So a lipid soluble or hydrophobic molecule won't be able to dissolve in the plasma very well, which is a serious problem if we just said that the endocrine system relies on systemic distribution through the plasma. So to solve that problem, we have carrier proteins in the circulation that bind to these steroid hormones and transport them where they need to go. Okay? In order to actually exert their effects, they have to dissociate from the transporter hormone, and then they're free to bind receptor. But you do need to know that as they're traveling through the circulation, they are bound to carrier proteins. But once they dissociate, because they're so hydrophobic, they can diffuse through the plasma membrane, which allows them to bind intracellular or intranuclear receptors. Okay? So they're going to have a little bit different mechanism of action. They don't need to rely on a second messenger to transmit the signal across the membrane. They can diffuse right across themselves. And as we'll see in a moment, these are going to be more focused on modulation of gene expression. Another thing, the peptide hormones can be bound by a cell membrane because they're hydrophilic. So they can be stored inside vesicles within the cell and secreted as needed. But with a hydrophobic molecule like a steroid, it will diffuse across membranes, so you can't store it in a membrane-bound vesicle. Because of that, these hormones are going to have to be synthesized as needed upon stimulation in order to exert the proper effects. So I say adrenal cortex secretes steroids. The skin is responsible for the production of vitamin D. The testes and ovaries and placenta also secrete steroids. And then the third class of hormones, which is the least numerous, is the amine hormones, which refers to amino acid-derived hormones. There's one that you need to know that is derived from tryptophan, and that's melatonin from the pineal gland. And as you may know, that helps regulate the sleep-wake cycle. So you can buy over-the-counter melatonin as like a sleep aid. It should help you with that. But the other important ones are going to be derived from the amino acid tyrosine. Okay, a couple important ones. The catecholamines, which consist of epinephrine, norepinephrine, and dopamine, are derived from tyrosine. Here's epinephrine over here. And the amine hormones are kind of interesting because some of them act like peptide hormones and some of them act more like steroid hormones. So you're going to be responsible for knowing which ones do which. So the catecholamines are going to act like peptides. They're going to be a little bit more on the hydrophilic side. Okay, so they're going to bind membrane receptors and transmit their signals through second messenger systems. On the other hand, the thyroid hormones, triiodothyronine and thyroxine, they have two tyrosines that are coupled together. Okay, so that extra ring makes them a little bit more hydrophobic, and it allows them to act more like a steroid hormone. Okay, so T3 and T4 act somewhat like steroid hormones. They're bound to carrier proteins in the plasma as they circulate. They dissociate to exert their function, and when they do, they can diffuse through cell membranes. They can bind intranuclear receptors, and like steroids, they're going to modulate gene transcription. Okay. So this one relies on a little bit more background information, but it's certainly a fair question. Okay, so I'm asking you which hormone is synthesized by the ribosome. So you need to know what the ribosome does. And the ribosome is responsible for the synthesis of polypeptides, or proteins. Okay, so there's only one hormone on this list that is a polypeptide, and that's insulin, choice B. Okay, something that may have confused you, epinephrine is amino acid derived, but remember, it's one amino acid. So the ribosome is responsible for coupling amino acids together to create a chain called a polypeptide. So if we just have one amino acid, like a tyrosine-derived hormone, it's not going to be synthesized by the ribosome. Okay, so as I say, the peptide hormones, all the rules that apply to any other protein are going to apply to peptide hormones. So they're going to be synthesized on the ribosome. They're going to have a signal recognition peptide that's going to localize them to the rough ER. They will be synthesized within the rough ER, and as they go, they will be modified as they travel through to the Golgi, where they're stored in secretory vesicles, and eventually exocytosed into the plasma, where they can be distributed and exert their function. Okay, so all of that information is fair game for the MCAT. I didn't teach you that. That will be in a different lecture, but you should be kind of keeping all of the different disciplines in mind as you're studying for this test. All right, so we'll move on to the mechanisms of hormone action. So we have two basic mechanisms, one major delineation. We have second messenger systems, and we have transcriptional modification. So as I've been saying up to this point, second messenger systems are most necessary when the hormone itself can't traverse the plasma membrane. Okay, so you need to somehow get the signal to the intracellular environment so the cell can do something about it. Okay, so we have a membrane receptor. Binding of the membrane receptor activates second messengers, and those second messengers eventually lead to the cellular response. And then a couple of key points to keep in mind about this is because you have multiple steps in this pathway, you have multiple steps to regulate the signal. Okay? And because you have multiple steps, you can also amplify the signal at each step. And we'll talk about that in a second. Okay? Second messenger systems are going to be fast-acting and short-lived, and they're going to be in response to peptides and the catecholamines. So remember, the catecholamines are the amine hormones that act kind of like peptides, epinephrine, norepinephrine, dopamine. On the other side, we have transcriptional modification, and this is when the hormone can traverse the plasma membrane and directly exert its effects within the cell. So we have intracellular or intranuclear receptors. The hormone is going to bind that receptor, and the hormone receptor complex, the two of them together, is now going to act as a transcription factor to target the transcription and the expression of certain genes. Okay, so because we're dealing with gene expression, okay, we have to make our messenger RNA, we have to translate our messenger RNA into polypeptides. This is going to be a little bit slower acting, but it's also going to be longer lived. Okay, so that's an important point to keep in mind. And then transcriptional modification, that's going to be the steroids that we just talked about, and the thyroid hormones, T3 and T4, which again are the amines that act more like steroids. Okay, here is an example of a second messenger system. The GPCR is testable material, so at some point you should learn in a little greater depth. But for us, it's just serving as an example of how the signal gets amplified. So if you were to be asked about one, you'd probably be asked about the stimulatory G protein, G-alpha S. So let's run through that pathway real quick. We have this 7-pass transmembrane receptor called the G protein coupled receptor. And then coupled to it, we have the heterotrimeric G protein. So upon binding to the GPCR, the G-alpha subunit will be allowed to dissociate from the beta-gamma. Okay, it will be able to diffuse along the membrane, and if it's stimulatory, it will activate an enzyme known as adenylate cyclase. And the G protein itself is active when it's bound to GTP, and it is inactive or turned off when it's bound to GTP. Okay, so if it is active and it's bound to GTP, it activates adenylate cyclase. And adenylate cyclase will then produce cyclic AMP from ATP. And this is one of the big steps of signal amplification. So one adenylate cyclase molecule can produce hundreds or thousands of cyclic AMP molecules. So in that step right there, you get a very large amplification of the signal. Okay, cyclic AMP will then go on to activate protein kinase A. And like other kinases, protein kinase A will phosphorylate various proteins, which alters their function and is responsible for the effect that is derived from this pathway. If we're dealing with other G proteins, we could have inhibitory, which does the exact opposite of stimulatory. So inhibitory is going to inhibit adenylate cyclase. And then we have one called the GQ protein, which has a completely different mechanism. And this one activates phospholipase C, produces IP3 and diacylglycerol, which eventually leads to increase in intracellular calcium. Again, that stuff is not essential for this lecture, but it's something you're going to have to learn at some point. And then the GPCRs are kind of the big class that you have to know, but don't forget about the others. So you can have enzyme-linked receptors, like insulin, for example, binds a receptor tyrosine kinase. So that will be more of a biochemistry lecture, but again, at some point, it's good to learn. Okay, so that was the second messenger system, and then we're working along, and this is the steroid mechanism, which is going to alter gene transcription. So we have a hormone here. The hormone is free to cross the plasma membrane. It's going to bind to its receptor, creating a hormone receptor complex. And this complex then acts as a transcription factor, binds DNA, okay, enhances transcription of target genes, messenger RNA, and then eventually into proteins. So this is a pretty good illustration, pretty simple of how this happens. And again, key points to keep in mind. The hormone has to cross the plasma membrane, and it's slow because it has all of these synthesis steps that need to happen. It's also longer lived. All right, so now we can keep moving on, and we're going to get into the regulation of hormones. And the big thing, the major concept in the regulation of hormone secretion, what's basically responsible for homeostasis in the body is negative feedback loops, okay? So pay attention to this, because you can memorize hormones all you want, and you can memorize their actions, and you can memorize what they are. But if you can't trace a negative feedback loop, and you don't understand the concept of negative feedback, you will be absolutely lost when it comes to endocrinology. Because this is where the money's at. This is what's going to tell you what's going on and what's going wrong when something is going wrong. Okay, so negative feedback. We have a stimulus. The stimulus leads to a response, and the response will then decrease that stimulus. And then when we decrease that stimulus back to where it should be, the normal baseline, that in turn turns off the response. So that is negative feedback. Here's the most basic example. Increased glucose is a stimulus for the release of insulin from the beta cells of the pancreas. As you probably know, insulin then leads to a decrease in plasma glucose, okay, uptake into the cells. And then that decrease in plasma glucose then stops the stimulus for more insulin secretion. Okay, that's very basic negative feedback. And that maintains blood sugar levels in the appropriate range, and that's a homeostatic function. And then we also have more complicated negative feedback loops that can operate on multiple levels. So what we just talked about is the response to the hormone ending the stimulus for the hormone. But when we get to multiple levels, the hormone itself can also inhibit the synthesis of other hormones. So the response can be negative feedback, and the production of the hormone itself can be negative feedback. So in this example, which we're actually going to talk about shortly, we have a substance, a hormone called corticotropin-releasing hormone in the hypothalamus, acts on the anterior pituitary to secrete adrenocorticotropic hormone. So adrenocorticotropic hormone acts on the adrenal cortex to stimulate cortisol synthesis, and that cortisol itself, as well as the response from the cortisol, but the cortisol itself can then feed back to inhibit both of these steps, which then, as you see, prevents the further synthesis of cortisol. And this is kind of the hormone saying, you know, there's enough of me, we don't need to synthesize any more for the time being. Okay, so there are your basic negative feedback loops. The opposite of negative feedback then is positive feedback, which is pretty uncommon, but there are a couple examples you might need to know. And being the opposite of negative feedback, positive feedback is going to be self-augmenting. So the hormone or the response to the hormone is going to provide a stimulus for the secretion of more of the hormone. And one of the main examples for this is in the menstrual cycle, the secretion of estrogen near the midpoint is actually operating under positive feedback, in that it increases the synthesis and release of follicle-stimulating hormone and luteinizing hormone from the anterior pituitary, which then leads to the synthesis of more estrogen, and so on and so forth. Okay, we'll talk about this later, so you don't need to write it down now. But the point about this is that it's this positive feedback cycle, and it will keep going to infinity unless there is an endpoint. So in a positive feedback system, there needs to be an endpoint. And in this example, ovulation is the endpoint. So ovulation becomes a stimulus to end the positive feedback cycle. Okay, so just a quick example, but this one again is much less common. Okay, and as you'll see, one of the big things about endocrinology as it applies to the MCAT is you're going to be asked to predict the levels of hormones. That's very important. And you're going to need to do this based on understanding the function they have and understanding how the negative feedback loops that they're operating in relates to the levels that you should expect. Okay, again, a very simple example. Elevated blood glucose should lead to increased insulin, decreased glucagon. We'll talk more about that later. So that's kind of the background you need to actually understand what the hormones are going to do. So let's talk about the major hormones. To begin, we're going to start up top, and honestly, this is going to be the majority of this lecture, just talking about the hypothalamic pituitary axis. Okay, so this axis is the master regulator of the endocrine system, basically. So, hypothalamus is structure in the brain, down to the pituitary, and the pituitary actually has two divisions, so we'll talk about those. Here's the pituitary. We have the anterior pituitary, okay? And the anterior pituitary is a true gland. It is true glandular tissue. That's important to know. And this glandular tissue, the secretion of its hormones is controlled by the neurohormones that are secreted by the hypothalamic neurons. Okay, hormones up here, traveling down to the anterior pituitary, modulating its function. And one major point we're going to make right here is that these neurohormones from the hypothalamus travel through something called the hypophyseal portal system. Okay, so this word hypophyseal, you may or may not have ever seen it. It comes from an old word for the pituitary, which is hypophysis, which literally means an outgrowth. So as you're looking up here, this pituitary is connected by a little stalk that connects to the rest of the brain. So it looks just like it's an outgrowth of brain tissue. So they called it the hypophysis. And they later named it the pituitary. So the hypophyseal portal system is going to transport the hormones that are made in the hypothalamus to the anterior pituitary where they can modulate their function. So let's take a little step back and talk about what a portal system actually is. Okay, so a portal system, definition, is two capillary beds in series. Okay, and if you don't know the difference between series and parallel, that's kind of like a circuits thing that you'll learn in physics. Most capillary beds are in parallel. Okay, so an artery branches out, branches out, branches out until it has all these capillary beds. and then they turn into venules and veins, and they come back together to form major veins. And those are in parallel. But when we have something in series, it's one after another. So we get an arterial capillary bed. That capillary bed comes back into a venule and a vein. But then that vein branches out into a second capillary bed. So that's two capillary beds in series. Okay, there are only a few examples, and one of them we're going to talk about right now. One of them is the hepatic portal system, which we'll talk about later. Okay, so the hypophyseal portal system. The neurons from the hypothalamus will secrete their hormones into one capillary bed. This capillary bed will travel in hypophyseal portal veins to a second capillary bed in the anterior pituitary. And there is a very important reason for this. Most hormones are secreted systemically, so they have high levels everywhere across the body. In this example, we really only want these hormones to be sensed by the anterior pituitary. So this allows the hypothalamic neurons to secrete small amounts of hormone that are sensed in high concentration by the anterior pituitary, because they're not being diluted by the rest of the blood volume. They're traveling directly to the anterior pituitary so they can have a large effect. Okay, so that's anterior pituitary. We'll come back to it. Posterior pituitary is the other half of the pituitary, and this is very different in that it's not a true gland. It is rather an extension of neural tissue from the hypothalamus. Okay, so this stalk of the hypothalamus, called the infundibulum, is actually composed, going to the posterior pituitary, of axons from the hypothalamus traveling down into the posterior pituitary. And the posterior pituitary is then simply composed of the axon terminals that come from cell bodies up in the hypothalamus. Okay, so we say it's an extension of neural tissue rather than a true gland. So what this means is that neurohormones are going to be produced by the cell bodies of the neurons in the hypothalamus. They are going to travel down the axon to the posterior pituitary, where they can then be stored and released from the nerve terminals in the posterior pituitary. Okay, a major conceptual division between the anterior pituitary and the posterior pituitary. And again, we'll talk about the hormones in a second, but first, a question. So the two hormones of the posterior pituitary are called oxytocin and vasopressin, and I want you to tell me how they actually get to the posterior pituitary. Okay, so again, we have a major division between the anterior pituitary and the posterior pituitary. So, as I was saying, posterior pituitary is an extension of neural tissue, and it's composed of axon terminals. Okay, now when we're synthesizing hormones, oxytocin and vasopressin being peptide hormones, which I suppose I should have told you, these peptide hormones need to be synthesized on ribosomes. They need to be packaged into rough ER and then Golgi and vesicles in order to exert their function. So all of the synthetic machinery in a neuron is going to be located in a cell body. Okay, so hormones of the posterior pituitary are going to have to be synthesized in their respective cell bodies, which are located in the hypothalamus. after packaging by the Golgi into vesicles, they can then travel down the axon to be stored in the axon terminals, and that's a process known as axoplasmic transport. Okay, so that is the walking of these hormone-storing vesicles down the axon to the axon terminal. For those of you who put the hypophyseal portal system, that is how the hypothalamus regulates the hormone production in the anterior pituitary, which is a true gland. Okay, so a major division there. Simple diffusion, it's a decent distractor, but I suppose a little bit of background information helps you differentiate between that. Okay, so there's my explanation. Again, synthetic machinery in the cell body packages these hormones and they then travel down the axon. So this is an illustration I like for the purposes of kind of demonstrating that the posterior pituitary is neural tissue. So you can kind of see these are axons running down this pituitary stalk into the posterior lobe. Okay, an extension of neural tissue, whereas the anterior pituitary is a separate entity and it's a true gland and it's glandular tissue. Okay, so sticking with the posterior pituitary for now. We just said that it produces antidiuretic hormone, which can be called ADH or vasopressin, and oxytocin. These are both peptide hormones, and let's talk about their function. So antidiuretic hormone is a very well-named hormone because it tells you exactly what it does. So diuresis means peeing a lot. So if you take a diuretic, you're going to be peeing a lot. Antidiuretic hormone prevents that. Okay? So it is going to, something we'll talk about in the next lecture, which is the kidney. It's going to increase the permeability of the collecting duct in the kidney to water, which is going to help the kidney reabsorb water and take water out of what will eventually become the urine. Okay, so we're decreasing the urine volume. We're preventing diuresis. It's antidiuretic hormone. Vasopressin, also a good synonym for ADH, because pressure in the vascular system, a function of ADH is going to be to increase blood pressure, as I say here. And something you'll also need to know is, and something we'll expand upon in the renal lecture, is that antidiuretic hormone is helping us reabsorb pure water from what will eventually become the urine. So if we're reabsorbing pure water, we're actually diluting the concentration of ions and various solutes in the plasma. So this is also going to act to decrease plasma osmolarity. And as you may be able to predict, just with negative feedback, the stimulus for the secretion of ADH is going to be a decrease in blood pressure. So ADH will then counteract that to increase blood pressure, or an increase in osmolarity, which then ADH can counteract and bring osmolarity back down. The other hormone of the posterior pituitary is oxytocin. And a couple things you need to know about this. It is involved in the stimulation of contractions during childbirth. Okay, it's another positive feedback hormone. So contractions are stimulated by oxytocin, but they also stimulate the release of more oxytocin. So you get more and more contractions until you finally have the childbirth. It's also responsible for milk letdown or milk ejection during nursing. That's about all you need to know about it. Okay, and we're moving to the anterior pituitary, which has a ton of hormones and a ton of functions, and it's very, very high yield. So there's a mnemonic I like to remember the hormones of the anterior pituitary, and it's flat peg. Okay, follicle stimulating hormone, luteinizing hormone, adrenocorticotropic hormone, thyroid stimulating hormone, prolactin, endorphins, and growth hormone. Okay, we're going to talk about all of these in more depth, except for endorphins, which are just kind of the body's natural painkillers. So the rest of them have various functions that we're going to talk about. Another reason I like this mnemonic a lot is because it delineates the tropic hormones from the direct hormones. So at this point, we need a definition to help us understand what a tropic hormone is versus a direct hormone. So a tropic hormone is a hormone that targets another endocrine gland. And that targeted endocrine gland can then produce its own hormones. So tropic hormones lead to the production of other hormones. Okay. As I was saying before, the hormones of the anterior pituitary are actually controlled by hormones secreted from the hypothalamus. So those hormones in the hypothalamus controlling the anterior pituitary would then also be tropic hormones. On the other hand, we have direct hormones, which have a direct effect on target tissue. Okay, so one example is the G in PEG, which is growth hormone. One of its target tissues is muscle, and it directly causes the growth of muscle tissue. It doesn't have an intermediate hormone between. So that is a direct hormone. Okay, so we are going to go through all these hormones and what they do. So buckle up. All right, follicle stimulating hormone and luteinizing hormone. They are secreted in response to gonadotropin releasing hormone from the hypothalamus. Okay, FSH causes the growth of the follicle during the menstrual cycle and the production of sperm in the male. Okay, that's more reproductive physiology, which is going to be a separate lecture, but as an introduction to that, that's the action of FSH. LH, on the other hand, you have a LH surge, which causes ovulation in the female. Again, that's reproductive physiology. But you also have secretion of sex hormones. In the female, that's estrogen. In the male, that's testosterone. Okay, so just a heads up on how this lecture is set up. We're going to talk about the hormones of the anterior pituitary, and then we're going to follow those hormones to the glands on which they act. So again, FLAT and FLAT, those are all tropic hormones, so they're going to act on other glands. So we're going to follow this. For example, we just talked about FSH and LH. So those are involved in the hypothalamic pituitary gonadal axis because they act on the gonads, which are the testes and the ovaries. Okay, so upon stimulation from FSH and LH, testosterone in the male and estrogen in the female will be produced. These are steroid hormones. Okay, pretty much a rule. If I forget to mention whether a hormone is a peptide, a steroid, or an amine, if I don't mention anything, it's a peptide. That's the vast majority of hormones are peptides. So I will always tell you if it's a steroid or an amine. Okay, so testosterone, estrogen, steroid hormones. They have effects in reproductive physiology, which is another lecture. But for the purposes today, we should just be aware that they are involved in contributing to secondary sex characteristics, which you are probably already aware of. All right, back upstairs. That was F and L. We're moving on to A, which is adrenocorticotropic hormone. It is stimulated to be released by corticotropin releasing hormone. And these names are pretty good. So corticotropin releasing hormone stimulates the release of a corticotropin known as adrenocorticotropic hormone. Okay, that's CRH from the hypothalamus, stimulates ACTH from the anterior pituitary. And then adrenocorticotropic hormone is tropic, meaning it stimulates the adrenal cortex, very aptly named. Okay, and upon stimulation of the adrenal cortex, we get the production of a glucocorticoid known as cortisol and androgen production in the adrenal cortex. So following that now, we are now in the hypothalamic pituitary adrenal axis, and we arrive at the adrenal cortex, which is stimulated by adrenocorticotropic hormone. So the major hormone of this axis is cortisol. Okay, again, it is a steroid hormone. This is kind of the long-term stress hormone of the body, whereas the short-term stress response is basically mediated by the sympathetic nervous system, which we talked about in the nervous system lecture. But the long-term stress response is in part mediated by cortisol. As I told you in the last slide, this is a glucocorticoid, and one of its major actions is gluconeogenesis, glycogenolysis, and lipolysis, which all contribute to increasing blood glucose. And that is why it's named a glucocorticoid. It increases blood glucose. So in this case, just so you know, ACTH secreting pituitary adenoma leads to the overproduction of adrenocorticotropic hormone. Okay. So the correct answer is B. Okay, so we have in this case the overproduction of adrenocorticotropic hormone. It is a tropic hormone that acts on the adrenal cortex, so it will directly lead to an increase in cortisol. Okay, and then that increase in cortisol will serve in a negative feedback loop to feedback on the hypothalamus and inhibit the production of corticotropin releasing hormone. Okay, normally an inhibition of corticotropin releasing hormone prevents the release of ACTH, but in this particular disease there's autonomous production from this tumor that is responsible for the disease. Okay, and there are tons of these diseases that you'll learn in medical school, but one of the main things about this is if a patient has certain symptoms that are related to an increase in cortisol, if you get enough tests you should be able to localize where the problem is. So you can say, with the proper test, is the problem in autonomous production of cortisol? Is the problem in autonomous production of ACTH, or even higher in CRH? Okay. So I know we're moving fast and it's tough to remember all these, but again, I'll make this stuff available to you so you can go through it at your own pace if you would like. But we got to keep moving. All right, back up top, our final tropic hormone. This is thyroid stimulating hormone. Okay, stimulated by thyrotropin releasing hormone from the hypothalamus. Thyroid stimulating hormone will promote the synthesis and release of the thyroid hormones, which are T3 and T4. So let's follow this to the hypothalamic pituitary thyroid axis. Okay, we have T3 and we have T4. These are the amine hormones that are more lipid soluble and tend to act more like steroids. Okay, so if you recall that mechanism, their action is going to be primarily focused on modulating gene expression, gene transcription. Okay, the function of the thyroid hormones is to essentially increase metabolism. Okay, so if you know anybody that's hyperthyroid or hypothyroid, they're relatively common diseases, the effects are directly related to the metabolic rate. So if you're hyperthyroid, you get things like tremors and you can't focus and you lose weight and you're sweating all the time and you can't stand to be in hot places. Versus if you're hypothyroid, you feel cold all the time because your metabolism is low, you start to gain weight even though you're eating the same, and various other effects. Okay. Okay, here's another one. So this is Hashimoto's thyroiditis, which you may know somebody who has. It's relatively common. Autoimmune destruction of the thyroid gland. So that renders the thyroid non-functional. Okay. Better. So, destruction of the thyroid. Okay, if the thyroid is destroyed, we can't very well make T4, which is one of the thyroid hormones. But because we're losing that feedback, we now are going to produce more thyroid stimulating hormone in an attempt to signal the thyroid to make more T4. Okay, the thyroid stimulating hormone doesn't know that the thyroid is destroyed, so it's going to keep making TSH, hoping that eventually you'll make more thyroid hormone. Okay, again, simple negative feedback gets you a long way. All right, so those were the tropic hormones, and we went through them very quickly. The main focus of that is that the pituitary and hypothalamus and that axis is kind of a master regulator of a lot of hormones. Okay, that's a big concept to understand on test day. As far as memorizing the functions, I can't stand up here and teach you memorization, so that's something you might have to make note cards and go over again and again until you understand them. Okay, but the concept of a hormone controlling another hormone and the concept of negative feedback are the points that I'm trying to get across to you in today's lecture. Okay, moving on to the direct hormones of the anterior pituitary. We have prolactin. Okay, prolactin allows for lactation because it is responsible for milk production. Okay, coupled with oxytocin, which is responsible for milk ejection, the combination of prolactin and oxytocin allows for nursing. Okay, you have milk production from prolactin, and then you have milk ejection from oxytocin. One of the really, really important things about prolactin, really high yield for exams, is that it is tonically inhibited by something called prolactin inhibitory factor, which is actually just dopamine from the hypothalamus. Okay, tonic in this sense basically just means constant. So it's constantly inhibited by dopamine. Okay, we talked about other hormones that are stimulated by the hypothalamus, but this is a little different in that without any stimulation from the hypothalamus, okay, it will functionally produce prolactin. Okay, it doesn't do that all the time because it's always under inhibitory control from dopamine, which is known as prolactin inhibitory factor. Okay, growth hormone. Growth hormone stimulated by growth hormone releasing hormone, and this is a pretty easy one. It basically allows you to grow. Okay, so a child with too much growth hormone will have something known as gigantism, which is they just grow too much. Okay, an adult with too much growth hormone, in the adult, the growth plates in the bone have fused, so an adult, you get more lateral bone growth and you get soft tissue growth. So you have like a really big tongue, you have dysmorphic facial features from lateral bone growth, but the main point about growth hormone is growth. Okay. Okay, so most of the hormones in the anterior pituitary are stimulated by tropic hormones released from the hypothalamus. And then the major exception to this rule is prolactin, which is, like I said, under tonic inhibition. So if you block the hypophyseal portal vessels with something like a tumor, okay, the hormones from the hypothalamus can't get to the anterior pituitary, which means prolactin inhibitory factor, dopamine, can't get to the anterior pituitary. Okay, so that relieves the normal inhibition on prolactin synthesis. Okay, so that was the anterior pituitary. Again, the master regulator, involved in a lot of tropic hormones, but it doesn't control everything, so there are some that we missed, and we're going to have to go through that now. The adrenal glands, okay, starting with the adrenal cortex. Okay, cortisol is basically under control by ACTH, but aldosterone is also produced by the adrenal cortex, and it has its own control. So aldosterone is a mineralocorticoid, which means it's responsible for regulating mineral or ion levels within the blood, as opposed to a glucocorticoid, which is involved in glucose regulation. So the mineralocorticoid aldosterone promotes sodium reabsorption and potassium secretion in the distal tubule and the collecting duct of the kidney, which we will go over in the renal lecture. This reabsorption of salts leads to a net reabsorption of salt, and water follows by osmosis, which helps to increase blood pressure. And this has its own system of regulation called the renin-angiotensin-aldosterone system, which will be in the next lecture. As a reminder, cortisol is the glucocorticoid, and then androgens are also produced by the adrenal cortex. You can remember this with the mnemonic salt, sugar, and sex, produced by the adrenal cortex. And those are all steroid hormones in the cortex. Okay, the adrenal medulla is something we talked about in the nervous system lecture, because the adrenal medulla is actually modified neural tissue, and it's involved in the sympathetic response. So it is responsible for the production of epinephrine and norepinephrine, which are the water-soluble amines that act more like peptides. Okay, and they mediate the sympathetic response. So something important that I said last time, the adrenal medulla, being neural tissue, is ectodermally derived, as other neural tissue is, whereas the adrenal cortex is mesodermally derived. Okay, something you might be tested on. All right, moving on. Calcium homeostasis. This is another big one. Okay, we talked about the thyroid and that it secretes thyroid hormone, and that's under control with the hypothalamic pituitary thyroid axis. But there are a part of the thyroid called the parafollicular C cells, C stands for calcitonin, that are involved in calcium homeostasis. Okay, and they produce calcitonin. And calcitonin tones down plasma calcium concentrations. Okay, leads to a decrease in calcium. It does so by promoting the mineralization of bone. So it takes calcium out of the blood and sticks it onto bone. It also increases potassium excretion through the kidney and the urine, and decreases calcium absorption in the gut. The antagonist to calcitonin comes from the parathyroid, which is next to the thyroid, and it secretes parathyroid hormone, which you can remember by phosphate trashing hormone. Because in addition to antagonizing calcitonin and increasing blood calcium, it also decreases blood phosphate levels. Here's how this works. It increases bone resorption to take calcium and phosphate out of the bone. Okay, but there's somewhat of a problem with this, because calcium being strongly positively charged and phosphate being strongly negatively charged, they form these crystals that help to mineralize the bone. But when you take them out of the bone into the blood, with the opposite charges, they're going to bind to each other very quickly. Okay, and calcium bound to phosphate isn't going to be functional in regulating blood calcium levels. So to correct for that, parathyroid is also responsible for the increased excretion of phosphate. So if you increase both calcium and phosphate, but then you take out the phosphate, you're left with free calcium that can exert its biological action. Okay, so increased bone resorption, increased phosphate secretion, vitamin D activation, which increases dietary calcium and phosphate absorption. Okay, and we'll end with one that's a little easier that you're probably familiar with. This is glucose homeostasis. Okay, so the control of blood glucose mainly comes from the endocrine pancreas. So the pancreas is both an exocrine and an endocrine organ. So right now we're talking about endocrine, which is hormone production. And the endocrine cells of the pancreas are known as the islets of Langerhans. They're composed of alpha cells, beta cells, and delta cells, which we won't talk much about today. So insulin, a peptide, is produced by the beta cells of the pancreas, and it acts to decrease blood glucose, mediating the uptake and storage of glucose into the other cells in the body. Glucagon is the antagonist to insulin, and glucagon acts to increase blood glucose levels. Okay, it is a peptide produced by the alpha cells of the pancreas, and it increases blood glucose via gluconeogenesis and glycogenolysis. All right, so that was a lot. I know that was a lot, and we went really fast. Okay, but in order to just get a feel for all these hormone functions, this is something that you're going to have to go over a couple times. But the main takeaways are the control of hormonal secretion being primarily mediated by negative feedback, and the ability to trace a negative feedback loop and predict hormone levels is going to be one of the most important things for endocrinology as it relates to the MCAT.